Hi, I'm Dr. Suraj Chirania, hematologist and BMT physician. Welcome to our channel. Today we will discuss genetic mutations in myelodysplastic syndrome from patient perspective point of view. What is the importance of this mutation? So, as we know, MDS stands for myelodysplastic syndrome. Myelodysplastic syndromes are the defective cells that are produced from the bone marrow. Usually, our bone marrow produces three types of blood cells: red blood cells, white blood cells, and platelets. In myelodysplastic syndrome, these defective RBCs, WBC platelets are formed from the bone marrow, that is a factory of blood cells. These cells are formed by mother cells that are stem cells. If stem cells has some mutation that give rise to these problematic cells, give rise to a condition what we call as myelodysplastic syndromes. So myelodysplastic syndrome, in a broader perspective, we called it as a precancerous condition because 1 in every 3 MDS patients can turn to acute myeloid leukemia in the future. How are these genetic mutations happen? Sometimes there are genes that constitutes the main part of DNA. These genes guide the normal functioning of the cells by various protein formed because of the coding presence in this type of genes. These genes, because of some drug chemical mutations or ionizing radiation exposures, become defective that give rise to these defective cells. But in some processes like normal aging, these genetic mutations build up over the period of time as the age advances, so leading to formation of these defective somatic mutations. So if we see, there are two type of genetic mutations overall. First mutation is a driver mutation. Driver mutations are the genes that leads to the defective growth of these cells. And second is a passenger mutation, means these mutations are present but they are not driving the growth and the existence of these mutations. Usually in one MDS patient there are 2 to 4 driver mutation present that leads to the growth of these dysplastic cells inside a body. So which are the tests we should do to know and understand the genetic mutation? First, most important test what we do is a karyotyping. If we understand what is karyotyping, it's just mapping out what are the different type of problems in the chromosome. In our body, there are 23 pairs of chromosome present. Chromosomes contains DNA and DNA contains genes. The defect in genes gives rise to defective proteins, which in turn leads to a defective cells. So in karyotyping, we map these 23 chromosome. Sometimes there can be shortening, lengthening of these chromosomes. Sometimes there is translocation, means one arm of one chromosome overlap on the other arm of the chromosome, leading to a translocation. There can be amplifications or sometimes duplications. These are all problems happens in the chromosome that can be easily detected by the karyotyping. Second, the smallest part of this chromosome is genes. There are a gene level test called as next generation sequencing. In short, we call it as NGS. In this genetic testing, we will identify the driver as well as passenger mutation. Sometimes these mutations are good or sometimes are bad. In low risk myelodysplastic syndrome, there can be mutation like SF3B1, which is a good mutation that respond to various drugs. While in high risk MDS, there are mutation like TP53, which is a very prone mutation that gives rise to acute myeloid leukemia in most of the patient which carries this mutation. So understanding these mutations is very important, since there is advent of targeted therapy, high intensity chemotherapy, or sometimes stem cell transplant These are the treatment option can be opted depending upon this mutation. In brief, if you plan to see the role of the genetic mutation understanding for the prognosis of myelodysplastic syndrome is very important, because it tells overall the prognosis and what is treatment ahead and what future lies for this patient with MDS. Thank you for listening to today's talk. Until next, stay informed, stay happy, stay healthy.